Let's talk about the qualities of an ideal delivery system. An ideal delivery system, first, which could accommodate a broad size range of inserted DNA, which could be targeted to specific types of the cells, which would not permit replication of the DNA, and which could provide long-term gene expression, and which could be easily produced, and which will be non-toxic and non-immunogenic. There are two major delivery systems for gene transfer. First, viral mediated gene delivery. Various viruses are used as carriers, for example, retrovirus, adenovirus, and adeno-associated virus, etc. The second group we call nonviral mediated gene delivery. There are various types of synthetic vectors are used as carriers, for example, cationic lipid, cationic polymer, etc. Now we talk about viral mediated gene delivery. Why virus is used as a tool for gene therapy or delivery. First, viruses have evolved mechanisms to enter into the cells. Second, they can replicate inside the cells. And third, they use cellular machinery to express their genes. But for viral vector to be useful, several viral functions must be altered. First, at a minimum, the virus must be rendered replication incompetent to prevent uncontrolled spread of the transgene. Second, some elements of its own genome must be removed to allow for insertion of transgene. Third, beyond this, additional modifications are dependent on the specific virus to avoid its toxicity. Take a look on common viral vectors. First, retrovirus vector, adenovirus vector, adeno-associated virus vector, herpes viral vector, and alpha virus vector. For example, these are the adenovirus vector. Adenovirus can enter the cell using the receptor-mediated mechanism. After entering the cell, the virus will be breakdown and releasing vector, and vector DNA will enter to the nucleus, so they can produce the new protein that have a therapeutic effect. And you can see the virus protein on the envelope can bind to the receptor on the surface of the cells before they can enter into the cytoplasm. These are retrovirus structure. These are common or basic retrovirus structure. This virus contains envelope or surface glycoprotein GP120 on the surface, you can see, and also envelope transmembrane glycoprotein TM, or we call GP41 here, and also contain GAG membrane associated matrix protein MA or P17, and also GAG capsid CA or called shell P24, and also RNA, 2 molecules of RNA this virus. You can see single-stranded, 2 single-stranded RNA, and also pol or protease PR, P9 or polymerase are reverse transcriptase, RNA integrase, for example P32. Yeah. This is the basic structure of retrovirus. How about non-viral vectors? An alternative to use of viral vectors for gene delivery is to deliver genetic material in the form of bacterial plasmid DNA. In the simplest form, naked plasmid DNA can be injected into skeletal muscles, leading to transfection of muscle fiber close to the site of delivery. Though the transfection efficiency by non-viral vector is relatively lower than that by viral vectors, synthetic non-viral vectors are designed to overcome many of the problems associated with viral vectors, such as risk for generating the infectious form or inducing tumor-genic mutations. Second, risk for immune reaction. Third, limitation to the size of genes incorporated and difficulty for production to scale up. The advantage of non-viral carriers over their viral counterparts are: first, they are easy to prepare and to scale up. Second, they are generally safer in vivo. Third, they do not elicit a specific immune response and can therefore be administered repeatedly. Fourth, non-viral vectors allow for the delivery of large DNA fragments and are also particularly suitable to deliver oligonucleotides to mammalian cells, which is excellent feature for the application of antisense strategy to down-regulate expression of certain genes. Fifth, they are better for delivering cytokine genes because they are less immunogenic than viral vectors. Take a look on major methods in non-viral systems. First, gene transfer with naked DNA. Second, gene transfer by physical methods. For example, we can use gene gun or electroporation or electronic pulse delivery or EPD technology or ultrasound facilitated gene transfer. Take a look on gene delivery by chemical methods. In chemical methods, various types of synthetic vectors have been developed for gene transfer. These include: first, cationic lipids, Second, cationic synthetic polymers. Third, peptides that act in a non-specific manner. And fourth, peptide and carbohydrate-based targeting ligand. Oligonucleotides. Oligonucleotides are short single-stranded segments of DNA. In general, there are about 13 to 25 nucleotides in length and hybridized to a unique sequence in the total pool of targets present in the cells. Due to their antigen and antisense application, they are used either as therapeutic agents or as a tool to study gene functions. Another example is intravene TM injection. Intravene is a sterile, aqueous, preservative-free bicarbonate buffer solution for intravitreous injection. It is represented by the following structure formula. This we can use to treat some of ocular disease. Intravene is indicated for the local treatment of cytomegalovirus retinitis in patients with AIDS who are intolerant or have a contraindication to other treatment for CMV retinitis, or who were insufficiently responsive to previous treatment for CMV retinitis. Take a look on siRNA or small interfering RNA. This siRNA is the effective method for gene silencing by introduction with double-stranded RNA into the cells. The mechanism of action is dependent on enzymes called dicer or RISC. The result is the degradation of target mRNA by RISC endonuclease activity. So, this is the end of the first lecture. Thank you for your attention.